since 2009 actually. We had a project looking at high temperature stress in sorghum, and I'm going to explain to you the physiological basis for it, what we can do about it, and the take-home message is that once you get temperatures are over 36 to 38 degrees, you'll knock the pollen off, and that'll happen particularly in the time from when the spike's about to come out of the boot. It's still very sensitive at that time, the pollen formation's happening. And I should clarify, the experimentation we do, we're changing the air temperature in a controlled environment. So it's 36 to 38 degrees being experienced by the head itself. Now in a field where you've got good transpiration, you've got good water under the crop, it might be a 36 or 38 degree day, you know, for a human. But if you're standing out there in a sorghum field, you might feel quite cool if there's a bit of water around. It's got a lot of cooling of about 4 degrees. So the plants may well be 4 degrees below that air temperature. So all of this work was done based on air temperature, and we've adapted for that when we've modelled it. But it's really important to think about that. The 38 degrees climate record doesn't necessarily mean that that's what the head's going to be experiencing, which is when the risk is going to happen. However, if that head is still inside the boot, it's likely that it can experience a higher temperature because it's isolated in the plant, it's not getting cooled as well. So I just wanted to make that clear at the start. The other messages are that we found that sorghum genotypes differ in tolerance to high temperature stress. The risk obviously depends on the sowing date and variety. We'll show you how we're looking at that. And we want to mention a little bit about climate change because you know, the temperatures have increased over the last 50 years, and our cropping zones have increased 0.5 to 2 degrees depending on where you are. But the more important thing is that a change in the mean temperature doesn't necessarily have a big impact for us, but an increase in the number of hot days does have a potential risk for us. That's the most frightening part of it, is the increase in the number of hot days. So we all know that every growing season is different. In the summer cropping area in the northern zone, we use variety and management to try to influence our yield, to optimise our yield, and we can use crop simulation tools such as APSIM to look at those risks. So we can simulate, we can take 50 years worth of data, we can look at, for any of those given years, with the profile of temperature and rainfall and the dynamics of all of that, how was the crop affected by stress and what was the impact for yield. So this is just to demonstrate that the APSIM crop model for sorghum is quite good. These correlations here from some published work we've done scientifically validating the model. It's perhaps our best crop model. It's had a lot of research put into the science that's in the plant itself, understanding that plant. We've spent a lot of resources making sure this is a very good model for the purposes of what we're talking about here. So the way we did this, at CSIRO, I haven't got a photo here, we have a thing called the Controlled Environment Facility, which is a part of our building at St Lucia. Inside that we have some rooms that are about 3 metres by 3 metres by 3 metres. We also have some much taller ones where we grow sugarcane and macadamias and things. But these rooms have a completely controllable climate system. So we can control the temperature every minute if we want. And it's very important because a lot of glasshouse or greenhouse experimentation that people do, they just have a daytime temperature and a nighttime, right? So they just set those two and let it go. For this kind of work, we need a lot more control. So what we've done is, you can see here, we've used the same night temperatures. We've ramped them up the same up until, I think, about 10 or 11 o'clock. And then through the middle of the day, we've mimicked different types of stress. So we've hit them up to 38 degrees in some of the experiments. And the reason that we've done it that way is because if we run them at 38 all day, you'll get pretty strange plants. And that doesn't really mimic what happens in the real world. We've tested 24 diverse sorghum genotypes out of David Jordan's breeding program or out of selections that he's got in the program. They're mostly lines, parental lines, not hybrids. And the first experiment that was done was looking at setting this maximum at either 30 or at 38 for the whole cycle. because that was the easiest way to just get the first set of data. And the experimental work here was done by Vijaya Singh and Chuc Nguyen, and Chuc as part of her PhD, and both of them were employed on this project, which was a DAF project, which I'll explain at the end. It was funded by the Australian government. So the kinds of things that they were doing was to look at pollen viability, for example. So if you shake the pollen off the plant into a little petri dish, you can then incubate that in agar, and you can go and count the number of viable pollen. It's a pretty laborious thing. Fortunately, you can do this in an air-conditioned lab. You don't have to do it in the 38 degrees. But it's a laborious methodology, but we needed to do this to validate what was going on in the plant. And then to look at seed set, at the end of the experiment, you're basically counting the number of grains that are on the spikes at different parts of the plant. And this is a comparison of a high temperature and a control for one of the lines. So the pollen viability in the seed set was reduced, but the genotypes differed. So this was that same variety, line B923296. You can see at 38 degrees, it's got almost no grains on it. And at 30 degrees, it's fine. So 30 degrees is pretty happy. But we've got another line here. So that's the same heads as here. Sorry, one of those are. This is the difference for 85G56, which is another parental line. And you can see at the high temperature, it's quite happy, whereas this one's not. So one of those heads is actually this head here. So there's a genotypic difference. And importantly, this pollen viability and seed set's really highly correlated. So if you look at the pollen viability and the seed set, it's very highly correlated. Sorghum is pollinating inside or very close to the grain. It's self-pollinated largely, so that pollen just falls down and pollinates the seed. And what that means is we can use seed set as a surrogate for this. So if we run an experiment like this, we can use seed set to estimate the effect. We don't have to do the pollen count. We had to do that to make sure that we were sure what was going on. So this graph, which I probably should have modified a little bit, it shows you a combination of the temperature and the genotypes. There's genotypes across here. Now, these are ranked in order of susceptibility to heat. So if you look at the 30 degree, which are the top bars there, at 32 degrees, about 80% seed set. So there's about 20% of the seed not setting, maybe for other reasons than the temperature. But if you look at this blue bar here, which may not be really clear, you can see for this variety here, or this inbred line here, we've got 5% seed set. So 95% of the seed is just not set at 38 degrees. And we've got another line over here that's got 60% seed set. So it's only lost 20 off the top of compared to the control. So there's real genotypic variation for this. The only commercial material that was in here happened to be MR Buster, which is sitting here at about 20% seed set. So it's been knocked around much more than some of these other parental materials. But just, I want to be very clear, this is not a test of hybrid materials. We haven't done that. We'd be quite happy to do it now that we've proven the method works, to get an idea of what the real range is in the commercial materials. So what we ended up doing was grouping them into 4 response types. If you look here, we've got the maximum temperature across the bottom, seed set here. So we're assuming it's 100%, and then when you've got a susceptible variety, it starts to react at about 33. So around about 33 air temperature around the head, it's starting to be sensitive for seed set, going down to 0 when it gets to 39. It won't have any grain at all. And for tolerant varieties, they don't really react until they get to around 37, and they are less susceptible. So it's certainly a big range. So we also wanted to check what's the basis for this, when's the timing important. And so Vijaya did a lovely experiment, which was a transfer experiment. So we had two rooms, one room that was at 30-degree max, one that was at the 38. And she was monitoring the plants every day. And once they got to flag leaf, she started shifting them from the optimum room to the hot room for 5 days. And then she shifted them back. And so what that shows very nicely in this case was that the sensitivity for seed set was at a maximum at 10 days after the flag leaf had appeared. So the last leaf had come up. If you put it into 5 days of heat stress, that was when you got the maximum sensitivity. So that's sort of just around the time that the head's about to come out or coming out. She also did the reverse, swapping them from high to optimum to show, that that was the time that actually made the conditions much better if you grew it in high temperature. I mean, if you grow it in high temperature the whole time, obviously you get quite low seed set no matter what. We also looked at the seed size, and the important thing with seed size is really the high temperature didn't directly affect that. It knocked it down a little bit, but it wasn't really sensitive to different timing. So unlike wheat, which is actually reasonably susceptible to high temperature just in that early grain fill period. So after that, we did some simulation work. I think you'll see in the, I think Moree was the example that's given in the book that you've got. I'm going to talk about the Gunnedah example. So we've implemented this response, so the slide that showed you how these genotypes are responding, we've implemented that into the model. We've put that in as a new algorithm so that we can run APSIM for, I think we used 50 years of data at Gunnedah and looked at the yield versus sowing date. There's a fairly flat response if you don't think about the heat response. So this is a model without the heat built into it. This is your median yield of about 3 tonnes. You're getting up around 5 tonnes in the top 20% of years and under 2 in the bottom 20% of years. And so there's not a really strong response to sowing date if you ignore high temperature effects. And what we want to know is there a response if you consider the high temperature effects? So all the modelling we've done so far and all the stuff we've published doesn't have this in it. So there's a little bit of, this is additional information that we haven't had before to be able to simulate this. So if we take a location like Gunnedah, here we've got probability of hot days against the time of year. So if you look at this top green line here for Gunnedah, what that's saying, or we can use Goondiwindi, that's where we are. If you're at Goondiwindi and you're in January, so in the middle of that plot, or December, January, you've got a 75% chance that you'll get a day over 36 in a 10-day block. So this is like a moving window, just saying how many days and what threshold you're going to hit. We just analyse the weather data to do that. And in fact, you've got a 25% chance of having 4 days over 36 in any 10-day block. And we're experiencing that right now. We're getting well over the 4 days, actually. And similarly, you can look at 38 degrees. So when you look at that against sowing day, I mean, we looked at the production risk without thinking about temperature. You go, well, it's pretty flat. In Gunnedah, you've got a pretty flat response to sowing date. But when you look at the risk of the high temperatures, up until when you're around this 16th of October, you've got a reasonably high risk of getting those 1 or 4 days, more importantly. So these dotted lines are the 4 days at 36 or 4 days at 38. So your maximum risk of being exposed to that sequence, not necessarily 4 in a row, but 4 in a 10-day period, is highest with these sort of mid-October plantings or even early October. As you go a bit later, you get into February, you're avoiding those heat risks. So what happens when we simulate this? Okay, so if we look at Gunnedah and you look at the relative effect of heat on a sensitive genotype, as we defined before, you can see quite a high proportion of years. So these are the different climate years. We've run this through the model. didn't put the mean in here. It's about a 20%, I think, maybe 20% loss due to heat in a susceptible genotype. So on that 15th of October sowing. So if you go for that middle sowing, you've got a sensitive genotype. The long-term median effect is around about 20%, 15 to 20% loss just due to heat. And it can be worse than that. It can, in the simulations we've got at the moment, you can lose up to 40% in a really bad year, and I think the personal experience of a few people is probably reasonably close to that. If you had a very high tolerance genotype, it is only 1 year out of the last 50 where it would have lost more than 10%. So most people, you would never see this. It just, that high temperature effect's going to be hidden inside pretty much everything else that you experience. We looked at the effect of sowing date and show how you've got a reduced risk with the earlier and later sowings. So this is just a plot of sort of all of the data together, and this is the relative yield. So you can see for those sort of early to mid-October, you've got a slightly higher median risk. But there's a risk there all the time. It's just that it's changing as you get earlier or later. No problem. So one of the last points I wanted to make, I mentioned this at the start, was climate trends. CSIRO's done a lot of work on looking at climate change, trying to predict with scenarios into the future climates. And I think the important message from here, I won't explain this in huge detail, but the main issue is that even a shift in the mean maximum temperature of 0.5 degrees or so for your summer period means that there's a great increase in the risk of exceeding threshold temperatures, high temperatures. So for this example, it's saying that by the time you get out to these, where are we looking? Okay, well this is from real data, in fact. So this is the changes that have already happened. And it's sort of gone, if you use a threshold, I think this one was of 36. Yeah, so between 1950 and 1980, the risk was about 2% in this monthly temperature anomaly, and it's gone up to about 10% that you'll hit these thresholds, that you'll get extra hot days. And I guess that makes this a little bit more urgent because we're just going to get more of these hot days. I forgot to mention one thing. All of the work that we did was under well-watered conditions, so those pots were well-watered. So these plants are in a good water situation, and in those climate chambers, we're managing the relative humidity perfectly so that we have the same VPD effect. We're not allowing the VPD to change with the two temperatures. So just to repeat the take-home messages, we can show clearly that high temperature around the head, 36 to 38, can be killing pollen in susceptible genotypes. The sorghum genotypes differ. We've been able to show very clearly there's a big genetic range, so there's a lot of potential that we can develop hybrids that can handle those higher temperatures. The risk obviously changes with sowing date and variety. We've looked at different locations. I gave you the example for Gunnedah, but the risk with the sowing date changes depending on where you are in the northern region. And there's certainly some concern that climate change exacerbating these high temperature effects. But the really good news, and physiologists don't always get to say the really good news, is that we know we can solve this. So it's probably one of the most satisfying pieces of research we've been able to do in the last 20 years because it was such a cut and dried case when we studied this. The mechanisms there, the genotypic variations there, we just need to put it into the germplasm and get it out into the industry. I'd really like to acknowledge very carefully the main research team who did the sorghum work, which is led by Graeme Vijaya and Chuc, who did most of the experimental work, with David Jordan's sorghum breeding materials, and Graeme, Erik, myself, my postdoc Bangyou Zheng, and Greg McLean at DAF. We worked with them on the analysis of the work and developed the new model and undertook those simulations. And I just wanted to mention that this was done as part of a federal government DAF-funded project. It was actually under the previous government who had funded some adaptation to climate change research. That research program unfortunately has been closed down, so there won't be any more money from that area. And it was partly supported by the funds that support Graeme's position at UQ. So thank you very much.